Oh, perfect. Let's see, is there going to be any of the participants with the webcam? Otherwise, I'm kind of just talking to nothing. I can't really see, who I'm talking to right now.
Yeah, Marty, Justin.
Is it recording? Oh, great. Oh, hello. Oh, okay. I saw somebody for like 2 seconds there. There you are. Hello. Hello. I don't hear anything from your side right now. All I do is, if I see you, I can't hear anything. Oh. Okay, that's good. Oh, all right. All right, sounds good. Fantastic. Are we still working on getting the visual back? Because for me, it's very strange as I'm talking just like to nothing. I can't see anyone. So for me, it would be really helpful if we could try again to get the video feed working. Oh, hello. All right. So everyone's here, sounds like. Okay. So I think Beverly is writing something right now. All right, perfect. So I think I'm talking to like 4 or 5 different classroom students right now, which is kind of crazy. So it's great for me to be able to talk to all of you students this morning, this afternoon, I guess. My name is Ann Meyer. I'm a professor of biology. I'm talking to 90 students. That's a lot. So I'm a professor of biology at the University of Rochester in the state of New York. So let's see, I've been a professor for about 8.5 years now. I started my lab, my first professor job was 8 years ago. I started my lab in the Netherlands in Europe. So I started my lab there for 7.5 years. And then I just moved back to the US just last summer. And so I've been, starting here in the US to bring some people with me and then hire new people again. So what does my job look like? So I do lots of different things during the day. Some things that I do are teach undergraduates, college students. That's just a really small part of the work that I do. So a lot of what I do is I run a research lab, which means that I have my own biology wet lab space where we have anywhere between 5 and 15 students working in it. So college students, sometimes high school students, PhD students, postdocs, research technicians, senior scientists, all working together. We work on lots of different projects, all the time, right? So when I first started my lab in Europe, one thing we were interested to do was we were looking at questions of how do bacteria survive when they're in a really stressful environment. So if they're starving, or if they're too hot, or if there's toxic chemicals in the environment, how can they survive? So we've learned a lot about that over the years. The most recent thing we've learned, I think, is so interesting because we discovered that when they're starving, especially, bacteria can literally, they condense their DNA into a crystal. So that's really weird, right? They have a crystal inside a living cell. But what's even weirder is that it's a liquid crystal. So it's packed like really tightly like a crystal, but it's constantly in motion such that some enzymes can get in there and express the genes. So these bacteria that crystallize their DNA, they can still express all their genes. So they can, make new proteins, they can try to fight back against the stress. So that's one thing we learned recently. That's kind of the fundamental project in my lab, so studying how does life work. We also have quite a lot of applied projects in my lab. So how can we use biology to make something useful or develop a new product, something like that, right? So these were things that I was kind of inspired to start working on when I was in Europe. I was in a technical university. That means that most of the people working there were engineers. So I got really inspired too to have kind of an engineering mindset to my biology. So we have several different kind of biological engineering projects. One is that we are using bacteria to make artificial seashells. So seashells are really amazing because they're stiff and they're tough, right? So for stiff, imagine something like a cracker, right? So a cracker, if you poke on it, it doesn't move very much. But most things that are stiff are not tough. So a cracker, if you try to bend it, it's going to break into 2 pieces, right? But then, another comparison might be, Jell-O. Like Jell-O, you can bend and it's not going to break, but it's not stiff, right? If you poke on Jell-O, it's going to loop, it's going to deform around where you're poking it. So seashells are amazing because they're stiff and tough, which is very unusual for material properties, which makes them great for something that is a structural material, like something you might use to build a building or to build an airplane or a car, something like that, right? So we use bacteria to make a version, an artificial seashell, so that we have a material that's stiff and tough. But because we're making it with bacteria, we can make it in a way that's really easy, really inexpensive, and really environmentally friendly compared to kind of chemical approaches to make materials. So we're making these artificial seashells. We're also doing these experiments to try to pattern bacteria, so to put bacteria in a certain shape, right? So what we're doing for this approach is my lab, we're building 3D printers. Maybe you're familiar with 3D printers that print plastic. So ours is based on a similar principle, but we're printing with bioink. So we're printing ink that contains bacteria. So with this approach, we can kind of deposit, lay down bacteria in a certain shape, and then we can engineer the bacteria to make a certain product or to behave a certain way. So, for example, one thing we're doing with this is we can print bacteria that are engineered to turn into biofilms. So biofilms are when bacteria start sticking together and sticking down to a surface. And they're really important because they cause a lot of diseases. Biofilms cause many diseases. So this way we can make like an artificial biofilm that has the same kind of shape and structure as a natural one, and learn more about how do these natural biofilms work. And then also we can use it as kind of a launching pad to make our own, engineered living materials, right? So biofilms, if they're not making you sick, if you get rid of the kind of part that makes you sick, they can be really useful because they're really tough and they're viscoelastic, right? So that means that you can't scrape them off a surface really easily. So we're trying to keep the viscoelastic and the toughness principle, but then we can add our own functions, right? So maybe we can make an engineered kind of biofilm inspired living material that purifies water or decontaminates water or something like that, right? So it could be like a reusable living water purification device. Yeah, those are most of the projects we have right now. So I would love to talk to you guys. So I'm really interested to hear, what questions do you have about being a scientist, about being a professor? Any questions about science in general, about what I work on? I'm here to answer anything that you guys want to ask. How do you make the bioink? All right, so when we're 3D printing, how do we make bioink, right? This is an important question. So we want something where the bacteria are going to be able to survive and be healthy enough so they can make a product, to make something, right? But we also want something where we can print it in the shape we want, but then once we print it, have it stay in that shape, right? So it's a lot of different things we have to think about. So what we're doing, how we solve this problem, at least for now, is so we have the bacteria, before we print it, is a liquid solution. So imagine just like a bottle full of liquid. It's kind of just like a bottle of sugar water. The bacteria loves to live in that. So it's growing and eating the sugar. And then we add to it a polymer, an unpolymerized polymer called alginate. So alginate, it's not polymerized, so it's also floating around in the liquid. And then, so we have a syringe pump. Syringe pumps are really great. They're literally, you like stick a syringe into a device that kind of pushes the plunger at a defined speed. So what that does is that pushes our liquid bioink through some tubing at a certain rate over to the print head where it drops onto the printing surface. Okay, but then we have, liquid bacteria we just printed, and that's not going to hold, it's not going to keep its shape after a couple of minutes have gone by. So what we do is we print it onto a surface that contains calcium. And when the alginate polymers in the ink, and the monomers, when that hits the calcium on the printing surface, it makes the alginate polymerize. So then our liquid bioink becomes a solid. So it solidifies and it forms this kind of like supportive scaffold that traps bacteria in place, but it's a hydrogel. This means that it's mostly water. So the nice thing with that is that food or chemicals or small molecules like that can diffuse, from the surface up into what we printed. So we can feed the bacteria and we can give them chemicals that tell them what to do next. That's kind of the principle of the bioink. Let's see, what question did we get? Yeah, exactly. So the question was, how do we get the bacteria to stay alive after we print the ink? Yeah, so alginate is kind of biocompatible. It's made by seaweed. So that's already a good place to start. And then we print it onto food. So if you grow bacteria in a lab, you kind of grow it on Jello. So you have like, this is like your plate. So you have like a dish and you pour some kind of Jello in there. So it's like, it's kind of like a jelly-like substance, and you've added some like protein and sugar in there. So then when you print your bacteria onto this Jello, then the sugar will diffuse into the bioink that you printed and feed the bacteria that way. So that keeps them healthy. And they're alive for at least 2 weeks after we print. I don't know, even longer possibly, but we didn't look for longer. Let's see. So there's a question from Molly Ludwig saying, how did I get interested in this field? So let's see. There's so many different things I got interested in at different points in my life. So the most recent thing I got interested in was when I made this, for me it was a big switch, to go from only studying fundamental science, to only studying how do living organisms function, to studying more applied science, doing more like engineering and making materials. So for me, that was all because of my teaching assignment in my first job. So when I started working as a professor in the Netherlands, my teaching assignment was I was running the iGEM. So iGEM is awesome. So iGEM, you should all do it if you're interested in this when you're in college. So it's a contest, undergraduates, college students, and it's not like a cutthroat contest. It's really friendly. The whole idea is it's teams of undergraduates that work together to try to engineer a new function into a living organism, right? And it is so cool because these students, they think of their own idea, they actually do it themselves in like, just a few months, right? From the start to the end of the school year. So when I was working with these students, they had all sorts of amazing ideas. We had ideas like, can we engineer yeast to sniff out, what was sniffing out? People are sniffing tuberculosis. We can detect diseases through a yeast that can smell. We've done bacteria that can detect the presence of explosive chemicals, like a bomb-sniffing bacteria. We've done, can we make bacteria coat themselves in glass to become like tiny little lenses? And I don't know, I was just so amazed at how creative these students could be and how creative these projects were and how useful the end results could be, that it inspired me to start my own work along these lines too. All right, there's a question. How do you engineer something in a living organism? So that is a great question. So yeah, there's really powerful tools now for engineering new functions in living organisms, but you have to kind of have the right approach. So, if you're thinking, this is like a way of thought that I use all the time at work, right? So what I'll do is I'll think about what's some crazy function that I want my bacteria to have, right? So for example, the glass coating, right? So let's say I want my bacteria to coat themselves on glass. That sounds impossible, right? The first thing I can do is I'll go to Google and I'll Google like organisms that make glass, right? Animals that make glass. And a lot of these kind of searches don't find anything useful. But when I Googled organisms that make glass, sponges make glass, some starfish make glass. All right, okay, so maybe this could work, right? Then I wanted to find out, how do they make glass, right? So some functions of organisms are really complicated, right? So like, let's say your function is, being smart, and there's like tons of, dozens of genes that contribute to being smart. And so you couldn't really easily transfer that function to a new organism. However, making glass is one single enzyme. So one protein encoded by one gene is all you need to make glass. So that was great because that's super simple. Because there are companies that can synthesize a gene for you, like in a test tube. So what we did was we found, okay, there's some sponge that makes glass, here's all of the DNA sequence that you need to make the protein, to make the glass. And then we give this like list, this like list of what the DNA sequence is, email that list to a company. They'll turn that into a piece of DNA that they made just for you. They'll mail it, through the mail to your lab. And then you can take DNA that goes into a bacteria. You can cut it, put your new DNA in, and glue it back together again. Like very literally. You literally cut your DNA, add your new piece, and then there's an enzyme that like glues the ends together. And you put that DNA into your bacteria, and that's really easy. You just give it like a very mild electric shock, and then it kind of the outside of the bacteria becomes a little bit porous, so the DNA can like float inside. And then when the bacteria like is feeling a little healthier after the shock, then the DNA that you made is in the bacteria, and it'll start making that protein, and the protein will make glass for you. That's kind of the basic workflow for one of these projects. Let's see. What are the applications of engineering bacteria to coat itself with glass? Right, so what could we do with the glass-coated bacteria? So maybe people don't know this, but micro lenses are like kind of the next generation of optical devices, right? So, traditionally, when you think about a camera lens, a telescope, something like that, you think of these lenses that are like pretty big, right? And those are very useful for certain applications, but they have a lot of drawbacks in the sense that to make them is not very environmentally friendly. It takes some complicated equipment at high temperatures and makes a lot of toxic waste, right? So it's already nice to make a lens in a way that takes place like at room temperature and doesn't make toxic waste and is really easy. That's already one advantage. And then if you can, instead of one big lens, if you make lots of little lenses that are the size of a bacteria, then, for example, you can put them on a flexible surface, right? So you could put them like on like a flexible piece of paper. So you have like a flexible array of lenses. And the smaller your lens is, so like these bacteria lenses would be the smallest micro lenses ever made, right? And so the smaller your lens is, the better it is, for example, capturing light. So if you have a solar panel that captures sunlight and turns it into electricity, then the smaller your lenses are, more efficiently you can collect light. So this is another example of why it's nice to have these little bacteria make the lenses for you, and it's really easy and cheap, and it will be the smallest lenses ever. All right, what other questions have I gotten? I apologize if I'm missing some questions. I only can see a few at a time in my window here. All right, let's see. I got the question. How long does it take for the bacteria ink to print? It's really, really fast. So the bio ink we make, it's not very viscous. It's not really thick. Because we print it as just a liquid, we can push it through our syringe and through our tubing really fast. So we can print a single layer in like a minute or less. That's really fast. What takes a bit longer is if you want to make, so it's like a two-dimensional structure. If you want to make a three-dimensional structure, so how does that work, right? So the first layer that you print, when you print it on a surface, calcium on the surface turns your liquid ink into a solid, right? And then you can print a second layer. And so how that works is, you go right on top of the first layer, print a second layer on top of that, but then that will turn into a solid only when calcium from the first layer fuses up to the second layer, right? So usually what we do is, after we print our first layer, we'll wait like, 3, 4, 5 minutes to make sure that enough calcium has diffused all the way through the first layer, so that when we print our second layer, there's enough calcium waiting, at the top of the first layer to make the second layer polarized. So yeah, so you have to wait like a few minutes between layers, but each layer you can print in less than a minute. So that's really quick. Let's see. That's a very concrete question. What is the average salary in my line of work? So being a scientist pays really well. So I recommend being a biologist for that reason. So it depends a bit on what exact kind of biology job you have. So if you're a professor of biology, there's different levels of being a professor. The starting salary of being a professor, depending on your university, might start around $70,000, $80,000 a year, something like that. When you're an associate professor, like a medium level, you might get $100,000. When you're a full professor, you might get $120,000, $130,000. So you make a lot of money as a professor of biology. Let's see. When you're, maybe you guys don't know this, but so when you are a student, right? So when you're a college student, you pay money to the college to be a biology student to get your bachelor's degree. But if you want to become a professor, after college, you should go to grad school to get your PhD. And in grad school, people don't often know this, but they pay you to get your PhD. This is really important, right? So almost anyone can get a PhD, after your bachelor's degree because they pay you to do it. They don't pay you a lot. They might pay you, again, depending on what part of the country you live in, they might pay you like $30,000, something like that, right? But it's enough, to live on comfortably while you're getting your degree. And then if you want to be a professor, so after your PhD, you'll become a postdoc. You get paid $5,000 or $10,000 more a year. Then if you get a professor job, you'll have a big leap in salary. Or you might have a job that you might work in a company, like work in industry, and you probably would get paid more than a professor that way. So there's lots of ways to make money if you have a degree in biology. Let's see. I've got a question. What kind of bacteria do I use? And do I get to name any new organisms? Gosh, do I name any new organisms? So I don't discover new bacteria. So people that discover bacteria get to name it. I don't do that. Plenty of people discover new bacteria. I could name it. I've never named anything, I don't think. So, for example, at this point, most of the genes already have a name, right? But some of the names are really bad names, right? So people went through and they sequenced they sequenced all the DNA in the bacteria, in most bacteria, right? So in thousands of bacteria, not most, in thousands of bacteria. And so we can look through all the DNA and we can look and see where the genes are. And then all these genes were given a name already. But if we knew the function already, we'll be given a name that reflects their function. If we don't know the function, they get just some random alphabet soup name. So if you discover a function for one of these unknown genes, you can name that gene a new name that reflects its function. The other thing people sometimes name is, for example, I mentioned that you can engineer a new function into a living organism. And if you do that kind of well enough, you can name, you can give an extra name to your species. So for example, I mentioned these iGEM teams, these college students that put new functions in an organism. I'm thinking of one where they took a bacteria species that make a lot of cellulose that's useful for all sorts of applications, and they engineered it so it could make even more cellulose, make more of it faster, and in a way that it makes it when you ask it, when you want it to make it. And then they named, they gave you the extra word, the name of the bacteria. There was I forget the name. It was something iGEM was the new name. And then I have the question, what kind of bacteria do I use? So I use whatever is going to be most useful for what I'm doing, right? So I'm looking at my fundamental research. So how do bacteria survive when they're starving or stressed? For that kind of research, I want to use the bacteria we know the most about. So the most well-characterized bacteria, we know the most about how it works, so that I have the most information and knowledge to build on in that situation. And so for that, the bacteria I use is E. coli. So E. coli, it's kind of famous as a bad bacteria because certain types of E. coli can give humans food poisoning. But there's lots of different types of E. coli, and the ones we use in lab don't make people sick. They're really harmless, and they're incredibly well studied. And E. coli is also usually what I choose if I'm putting a new function into an organism. Because again, we know how everything works, and we know really well how E. coli regulates its genes. So how does it turn on or off the genes? So if I'm putting in a new function, if I want a bacteria to coat itself in glass, I want it to do that when I want it to do it, and to express the genes at a certain strength, right? So we have all these tools. We know all of these DNA sequences that control when and how much a gene is turned on. So we can use those in front of our gene of interest, put it into E. coli. We know that it's going to work in kind of a reliable manner. However, there's lots of situations where I want to work with some weird bacteria that people found out in the environment. So, for example, I mentioned we're making seashells with bacteria. So there's a couple of steps when we make our seashells. So seashells are basically like tiny nano layers of cement sandwiched together by some sticky kind of silk-like molecules. So I have one species that people found in a puddle outside that can make layers of cement. It changes the pH of the solution to make calcium carbonate crystals form, so most of these little layers of cement. And then I have to sandwich these layers together with some sticky silk-like molecule. So for that, I use some bacteria that people found on bird feathers that happens to make some sticky polymer. And we don't really know why it does that on the bird feathers. But from my point of view, I don't really care why it does that. All I care is that it makes a lot of some sticky polymer. So I use that to like glue together by layers of cement to make the eggshell. Let's see. So how long have I been working with bacteria? So, I haven't used it for my whole career. So as a PhD student, I guess I first researched it as an undergraduate, as a bachelor student. I worked with all sorts of different systems then. I worked with yeast, various bacteria species. As a PhD student, I worked with cows. So I was purifying a protein from a cow to see how it works, because the cow protein was almost the same as the human protein that we were interested in studying. But we could buy some beef and purify the protein from the beef and get a lot of it that way. And then so that was kind of looking at animal proteins and how they work. And then only as a postdoc, so kind of like late in my career, only as a postdoc did I start looking at bacteria. Mostly because it's kind of a pain to work with the cow protein because the certain protein we were looking at was a very interesting protein, right? It was a protein kind of shaped like two barrels stacked on top of each other, like back to back. And these barrels were super cool because they were in charge of making sure that newly made proteins in the cell have the right shape, right? So proteins are made as like a long string of amino acids and turn from a long string to a three-dimensionally shaped final protein, right? And some proteins can't do that by themselves. And those proteins would literally get stuffed inside this barrel. And this protein was incredible because this protein has a built-in lid, but most of the protein is stuffed inside the barrel. This lid closes, so it traps this poor protein inside where it folds with the help of the barrel walls. And then after 2 minutes, the lid opens. That's a long time by the proteins. 2 minutes is a long time to wait for something to happen for a protein. The lid opens, and the bacteria can float away, and hopefully it has final shape. So I thought the protein was so cool that I went to all the trouble to purify it from some beef that I bought from literally straight from the butcher. But while I was doing that work, I was jealous of my friends that worked in bacteria because they could just make the protein they wanted to work with whenever they wanted to make it, because it's much easier to purify bacteria proteins. And plus, since they can make the protein in bacteria, that means they can engineer the bacteria to make different types of the protein, right? So let's say, for example, this protein that has the built-in lid, right? Let's say you want to study what's the importance of the lid. In bacteria, all you do is take the DNA that codes for this protein and you chop out the part that makes the lid. And then your bacteria make that protein, now you make the protein without a lid, and you see how it works, right? And that kind of experiment is very easy to do in bacteria, where you can, take a DNA of interest and put it in the bacteria and have it, be expressed as a protein. The cow tissue, you can't do that in cows, right? You can't take, or maybe now you're just being able to with CRISPR techniques, but for almost all of human history, you're not able to make a cow that makes a different kind of protein. You can't really change the DNA of a cow very well. So I wanted to work in bacteria as a postdoc because I thought I would have all the power to, change protein and study how that changes their function and add new functions. And so far, I've been really satisfied with my career choice to work with bacteria. Okay, there's a question about, are there any drawbacks or negative effects or long-term effects of any of these bacteria to do different things? Right, so there are always tons of consequences to anything you do to a living system, some of which you can anticipate and some of which you can't anticipate. So we always try as hard as we can to anticipate what's going to happen, right? So on a very kind of biological level of detail, one thing that can happen is bacteria don't often like to have a new function. And when I say like, they don't have emotions, right? But so bacteria divide really fast. They divide exponentially, right? So you start with one bacteria in the morning, and by the next morning you have like millions of bacteria because they divide, those divide, those divide. So anything that makes a bacteria divide more slowly is going to have a huge disadvantage in the population as a whole, evolution on a small scale, right? So like it's pretty common that like a new function you give to your bacteria is going to make your bacteria divide more slowly or even like stop dividing, in which case it can easily be swamped out by bacteria that don't have this function. So like imagine, go back to this bacteria that makes the glass idea, right? So when the bacteria make glass, they stop dividing, right? Because they can't divide through this glass shell there. They stop dividing. So imagine some bacteria in your culture, they lost the ability. They mutated their gene, they can't make glass anymore. So those bacteria are going to divide, as fast as they can every 2 minutes, right? And so if you let your bacteria culture make this glass, make the glass for overnight or for 2 nights, if any of these bacteria have lost the ability to make the glass, they're going to have overwhelmed and swamped out the ones that make the glass. So you always have to be aware of evolution happening in your bacteria cultures because that can really mess up your experiment compared to what you wanted. And then, it's really important to think about, let's say you have a new function in your bacteria and it works exactly how you want it to work. Let's say, it makes a new function you think is going to make a great product, make a great material that can go out into society, leave the lab, go into society. And it's always important to think about, would there be any ethical considerations, any economic considerations, something like that, right? So, for example, a lot of bacteria engineered to have a new function. The way you know if the gene that you put in there is actually in there is that attached to the gene you're putting in there, you put an extra gene that makes your bacteria resistant to antibiotics. And this is really powerful, right? So let's say you want bacteria to make glass, the same example again. So you pair it with a drug resistance gene and put the DNA into your bacteria, and then not all your bacteria will get your gene of interest. So then you add your drug to your culture of bacteria, and that kills all the ones that didn't get the new DNA or where it's not working, right? So then, all the ones that survived the drug will be the ones that have the new DNA that can make the glass. But then you have to worry about, if your bacteria would leave the lab and go out into society, you don't want this drug resistance ability to spread, right? We don't want bacteria that live on your body or in your body to be drug resistant, because then, if they make you sick, you can't kill them with drugs anymore. So, this is one example, right? Something where it's important to think about these kind of things. So, for example, for our bacteria that make the glass, they make little, microlens around themselves. For this project, what we do is we put these lenses under a strong UV lamp, ultraviolet radiation, that will kill the bacteria inside the glass, that will kind of shred the DNA. You don't want any of that DNA, any of those cells to be alive anymore once they leave the lab. And so hopefully, that would make it safe and acceptable to society to then be part of the product later on. Let's see. So what influenced me to pursue biology? Let's see. I think biology is just super interesting. One thing I remember, so the pivotal moment, was when I was a junior in high school. This is going to sound super dorky, but we had a really good textbook for my biology class in high school. Because this textbook was great because, for everything that it would tell you that we know about biology, it would also tell you all the things we don't know about biology. So they would tell you, I don't know, and they would talk about the specific scientists that had made the discovery. So-and-so discovered, how you can turn a gene on or off, but what we don't know is, almost everything else about it, right? So, every page of the textbook would just have some questions or some open-ended ideas that we didn't know about. And it got me so excited because there were all these things that were so interesting that seemed like someone needs to go figure out what they want, right? So it got me really inspired to be one of these people that, learns about the open questions in biology. All right, so a bunch of questions have come up from the Owens classroom. I'll answer maybe the simple one first, which is, do I travel any with my work? So so science is a great job if you like to see the world. And you don't have to if you don't want to. But so, for most of my job, what I'm doing is I'm, in my office, in the lab, in the classroom here in Rochester. But I travel all the time. I travel maybe at least every 2 months for all different reasons. So there's a bunch of scientific conferences that I go to. So this is where, everyone working in a certain field will come together and share their science and get to know each other and share techniques and share, chemicals, something like that, right? Share laboratory equipment. And those are really fun, and those are all over the world, right? So there's tons of conferences in America, a lot of conferences in Europe, Asia, just all over, right? I also travel, for example, I'm going to London next week because I'm part of a, how can I describe it? I'm part of a working group that is trying to regulate biosecurity on a kind of global level, right? So we're trying to make sure that people that do work like I do, that work on bacteria, work on viruses, engineer new functions in the virus and bacteria, trying to make sure that they always do their experiments in a way that is safe for society. So this is the panel with people from, countries all over the world, people that are in bioethics, in policy, in the government, in regulatory agencies, and people that are scientists are getting together to work on making new guidelines, new incentives for biologists to be safe and secure. So that's another reason why I'm traveling, right? I'm traveling to London to meet with all these different people to work on biosecurity guidelines. And there's tons of other reasons, when students defend their PhDs, when they're getting their PhDs, you always have an outside person. So I just, 2 weeks ago, I traveled to Europe to be on the PhD defense of a student. So there's tons of great reasons to travel when you're in science. All right, so my question, how did bacteria make the liquid crystal? So this is something I think is such an unexpected, interesting result that we found. So it's only due to one protein, which is even weirder, right? So I thought it would be a very complicated process. But, so here's kind of the basic idea, right? Is so when bacteria are not stressed, they have plenty of food, they're not exposed to toxic chemicals, their DNA is kind of loose. 2 more minutes, I'll wrap it up. So it's kind of like, some spaghetti or whatever in a cell, aligned in a certain way. And, already to shove enough DNA into bacteria cells is already a challenge, right? So it's wrapped around certain proteins that organize the DNA. And then when bacteria cells become stressed, one of these proteins that packages the DNA, they make tons more of it. So this one protein goes from not really common to being the most common protein on the DNA. And then this protein, when there's so much of it, it crystallizes. It's kind of like, salt crystals. Like you have a lot of salt in your drink, it'll form crystals that drop out of drop out of the water into the bottom of your cup, right? So when there's tons of this protein, the protein starts to crystallize. But at the same time, it's still binding the DNA. So that like draws the DNA into this crystalline complex that's held together by these protein-protein crystal interactions. All right, so are we out of time? It looks like we have 1 minute left, so I don't know if I can go any farther. Well, I hope that you guys enjoyed my talk today. Enjoyed, you know, asking and answering questions together. So it's great to talk to you guys, and don't hesitate to email me if you have any more questions in the future. All right, I'm glad you guys thought it was interesting.